This presentation is by McCall, Macahdo, and Madison Hall. It is on the topic of xenotransplantation, and we are against it. So, what is xenotransplantation? Xenotransplantation is a transplantation of living animal cells, tissues, or organs from one species to another. In this presentation, we will specifically be talking about the cons of organ, cell, and tissue transplant between animals and humans. According to the FDA, which is the Food and Drug Administration, also known as the U.S. FDA, is a federal agency of the United States Department of Health and Human Services, is one of the United States federal executive departments. So anyways, according to the FDA, the development of xenotransplantation is driven by the fact that the demand for human organs for clinical transplantation far exceeds the supply. Therefore, doctors and scientists are trying to further develop the success of xenotransplantation in order to meet the demands of needed organ transplants. In the U.S. today, 10 patients die each day while on a waitlist to receive a vital organ transplant, and I am here to show you the greater risk for these patients if they were given a non-human organ. So in the next slide, we will be showing you a video on xenotransplantation. So now we're going to talk about the history of xenotransplantation. In the early 1900s, doctors attempted to replace failing human organs with organs from animals such as pigs, goats, lambs, or monkeys. All of these xenotransplants failed, and any further attempts were abandoned until scientists had discovered why the transplants were failing. Non-human primates such as chimpanzees and baboons were originally used as xenotransplant donors, but concerns about spread of infectious disease and ethical issues have now stopped their use. Most researchers are now using pigs as donors because they are easy to breed and have large litters. Pathogen-free pig breeds are available. Pig organs are similar size to human organs. The risk of infectious diseases is lower than non-human primates, and pigs are already killed for food, so using pigs may raise fewer ethical concerns than non-human primates. So here is a timeline of xenotransplantation and organ transplant amongst humans. In 1902, they reconnected blood vessels for organ transplants. A man by the name of Alexis Carrel at the Rockefeller Institute in New York describes how blood vessels could be reconnected in transplanted organs. Carrel receives the Nobel Prize for his work 10 years later in 1912. In 1902 through 1923, the first attempts at organ xenotransplants happened. The transplants were with pigs, goats, and sheep. Sheep and monkey organs are attempted, but all fail, with patients surviving only hours or days after transplantation. In 1944, they discovered the immune system causes transplant rejection. A man by the name of Peter Medawar from the University of London discovered that transplants are failing because of an immune system reaction/rejection. In 1954, the first successful human-to-human transplant happened, and this was with the kidney between identical twin brothers. In 1963 through 1997, animal organ transplants in human bodies continued. During these years, xenotransplantation was attempted with organs from bamboos, chimpanzees, and pigs. However, there was little to no positive results through this, seen as patients would not survive long after the procedure. This slide is a continuation of the timeline of xenotransplantation and organ transplant amongst humans. So in 1997, pig liver was used to keep a patient alive. Robert Pennington, a 20-year-old with liver failure, was kept alive by passing his blood through transgenic pig livers which had been genetically modified so they would not be recognized by the receptant's immune system. This procedure is carried out for 7 hours and over 3 days until a suitable liver becomes available. This procedure is done a few weeks before a worldwide ban on xenotransplants. In 1997, the worldwide ban on xenotransplantation happened. Concerns about the risk of infecting human receptants with animal endogenous reservoirs lead to a worldwide ban on animal-to-human transplants. Pig endogenous reservoirs, also known as PERV, was of particular concern. In 1997 through 1999, the risk of infectious disease is assessed. Several groups published findings showing no evidence of the PERV infection in human receptants of pig tissues. In 2000 through 2011, the ban on xenotransplantation is lifted in some countries. The ban on xenotransplantation is lifted in some countries, and applications for trials with xenotransplants are assessed based on a case-by-case basis. So here's some ethical aspects of xenotransplantation. People believe that using animals to supplement human life is basically using them for our own needs. While it is highly accepted to use animal organs as another process in producing food, growing animals just for organ harvesting, disregarding their meat, would be a moral issue that society tends to hold. Also, people oppose xenotransplantation because of religious views that humans are superior to humans, therefore making them strongly uphold that it is against God's will. Here are a few disadvantages of xenotransplantation. So, a major disadvantage of xenotransplantation is the risk of rejection, and it is also known to have a high rejection rate. This is when the receptant's body attacks a new organ, like an infection. It is the greatest obstacle to undergo during xenotransplantation. The breeding of transgenic pigs, as well as new cloning techniques, may be used to reduce the risk of organ rejection. There are three types of rejection. There is hyperacute rejection, acute rejection, and chronic rejection. Hyperacute rejection occurs rapidly, often before the surgery is complete, as pre-existing antibodies from the immune system react with the donor tissue. Acute rejection occurs in the first weeks or months after surgery, as an immune system response develops, and chronic rejection occurs gradually over several years after surgery. Here are a few more disadvantages of xenotransplantation. Another disadvantage of xenotransplantation is the risk of disease, especially xenosis. Xenosis is an infectious bacteria transmitted from animal to human by the transplantation of an animal tissue or organ into a human body. Transmission of viruses from xenografts into humans. The possibility of xenosis raises questions about the safety of xenotransplantation and could also potentially place the general public at risk by creating a major new epidemic. Also, animals have much shorter lifespans than humans, which means that if the success rate improves for transplanting animal organs to humans, there will still be a risk of the organs wearing out or dying prematurely. This would also mean that a person would need to undergo multiple transplants over his lifetime, as the organs would be wearing out. So in the next video, we'll basically go over more cons and detail of xenotransplantation.
Now this is Macahdo, and I'll be talking on the sociological problems of xenotransplant. Hundreds of people have benefited from the advances made in organ transplantation. But xenotransplantation is lacking on social science compared to the scientific research and ethics of it all. Xenotransplantation has many major problems, such as animal rights and welfare, or another problem is whether humans want animal organs inside of them or not, and all the risk that comes with xenotransplant. Xenotransplantation is one of the newest issues in ongoing social movement protesting the exploitation of animals for human use in a way that is cruel and inhumane. The animals that are subjected to be used are caused serious suffering. This suffering is caused by the social context of the research. The research is ran by research groups who see humane demand on the treatment of animals as an obstacle, not a moral necessity. The donor animals suffer from the conditions they have to live in to provide safe organs for humans. They are isolated, which causes emotional suffering, especially to animals like pigs, who are very social animals. But why is this a public matter? One of the biggest problems with xenotransplant is in fact the risk of disease transmission to third parties. This is a public matter because xeno raises questions about justice through the possibility of creating new and dangerous diseases. People are scared of this because this could infect the general population. The main problem is that xeno risks infecting people with viruses that we are not equipped to take care of. From donor animal into organ recipient, from there these diseases could spread onto other people in the population. In addition, a number of factors make the risk at large of spreading serious disease to the general public, or at least theoretically it would. To continue on with the risks, there's additional risk factors where doctors are now require transplant recipients' immune systems to be suppressed. In other words, they want their immune systems to be lowered so typically in human transplants they would have a higher success rate of adapting to the new organs, but with animal tissues inside of them, the body might see as an alien and would reject it. But having a suppressed immune system encourages bacteria and viruses to happen. So having animal organs use the surface for xeno with human genes, then they will have a greater chance to become infected. That would make it harder for the human immune system to identify the organ as a good thing and accept it. So where are we today? Despite all the social efforts to increase the ability of organs from cadavers to living organs, it remains highly unlikely that the human organ graft will fulfill what is needed. But scientists are now saying their new research will allow them to breed animals just to harvest the organs. There are still challenges with this because of all of the ethical objections to using animal organs for human use. So what are some current events? Scientists have created the first human-pig hybrids to create the ultimate supply of organs for transplants. In Britain, today they are facing a huge organ donation crisis. Their demand is higher than their supplies. They have learned that even though pig organs are around the same size as humans, that doesn't change the fact that our immune systems will keep rejecting the animal tissue inside. So, who created the first human-pig hybrids? A team at the Salk Institute for Bio Students in the U.S. have put both concepts together and learned it is possible to grow human tissue in a pig. It took the institute over 4 years, 1,500 pig embryos, and then 40 human stem cells to create this. So how did they do this? First, they created a vacuum in a pig embryo by editing the pig's DNA. They then followed this by introducing the stem into the embryo. Third, they developed organs exclusively from human stem cells. And then they ended this by transplanting organs into humans from pig fetuses. This is our presentation on xenotransplantation, and we are against it. Thank you for listening. Have a good day.